Bismillahirrahmanirrahim. Assalamualaikum. Good morning, everyone. I would like to thank to you for coming to my presentation today. Today, I'm going to share with you part of my research, which is mitochondria protective effects of nicotine on hippocampal neurons of rapid eye movement, sleep deprivation, induced stress in the rat models. My name is NORLINDA binti Abdul RASHID, and I am from Kuliah of Nursing International Islamic University, Malaysia. Well, rapid eye movement or REM sleep is one technique to induce depression in animal models. It is referred as a highly activated cholinergic system in the brain in a paralyzed body. This is the stage of sleep by which information obtained during wakefulness are formed in the hippocampus and known as learning and memory. REM sleep deprivation can cause cell injury. This is because previous studies show REM sleep deprivation has given rise to reactive oxygen species or ROS and lipid peroxidation in the hippocampal neuron. When antioxidant activity is insufficient to balance oxidative stress, the cellular injury will occur. It was reported that high reactive oxygen species accumulation will overwhelm mitochondria, thus resulted in the injury of the mitochondria and furthermore the cell as a whole. Mitochondria is a multifunctional and dynamic organelles deeply integrated into cellular physiology and metabolism. Disturbance in mitochondria function are involved in several disorders such as neurodegeneration. Therefore, it is clear that sleep deprivation is able to induce mitochondrial damage of the cell. Nicotine is a natural alkaloid discovered in Nicotina tobaccum by Posselt and Raymond in 1828 and first synthesized by Pittet and Rothschild in 1904. It is produced in the roots of plant and accumulates in the leaves, and is also found in smaller amount in other members of Solanaceae family such as potatoes, tomatoes, eggplants, and chili peppers. And then it was reported that nicotine could be beneficial. Indeed, there is evidence for nicotine having anti-inflammatory and also anti-apoptotic property. In terms of the experimental design, 10-week-old male Sprott dolly rats weighing 200 to 250 grams were divided into a normal condition, depression model of REM sleep deprivation or R, and control wide platform group, which is W. And then it will further divided whether it is nicotine treated or non-nicotine treated. The rats were then euthanized with an overdose of sodium pentobarbital, and body was fixed using transcardiac perfusion fixation technique. The hippocampi was harvested and processed following transmission electron microscopy protocol and viewed under transmission electron microscopy. The change of mitochondria in the hippocampus was then scored using ultrastructural changing scoring system. So for the result of this study, it shows that in the hippocampus, there was significant difference seen between groups in relation to the changes seen in the mitochondria. In figure A, you can see that the mitochondria is actually from the normal hippocampus. So we can see this normal structure. B shows the mitochondria from REM sleep deprivation hippocampus showing swelling and damaged mitochondria with collection amorphous material, and C, mitochondria from REM sleep deprivation hippocampus treated with nicotine, and we can see the crystal looks prominent with a big swelling. However, it is not yet damaged. So the quantitative point of view, a REM sleep deprivation group due to REM sleep deprivation show the highest score of changes in hippocampal mitochondria, and this score was significantly different when compared with other group. As a discussion, although the degenerated neurons cannot be detected earlier than 6 days of REM sleep deprivation when the aminocupric silver technique and light microscopy is used, 24 to 96 hours of REM sleep deprivation give rise to increase of ROS and lipid peroxidation in the hippocampal neuron, which indicate that neuron progressively undergo ultrastructure changes, which can be detected using high magnification such as electron microscopy. A mitochondria is an organelle that is responsible for producing ATP through oxidative phosphorylation in the electron transport chain or ETC. REM sleep deprivation can impair the ETC ADC, leading to mitochondria dysfunction and increased ROS production. When ROS accumulates, it will overwhelm the mitochondria and cytoplasmic antioxidant system, hence ultrastructure damage will occur. Therefore, in this study, the detected ultrastructural damage could be due to the accumulation of ROS following the failure of antioxidant system to counteract the effects. Nicotine showed the exact beneficial and protective effects in few neurodegenerative disease. This effect is consistent with our finding in which the ultrastructure of the mitochondria in RN group show prominent vacuolated cristae. However, this appearance was in a better condition if compared to the REM sleep deprivation group. In addition, the reduction of ROS production by acute nicotine treatment ameliorated the REM sleep deprivation-induced ultrastructural damage in the hippocampal neuron. Thus, we suggested that based on the hippocampal ultrastructure or mitochondria score in the RN group, there was a significant improvement. For conclusion, REM sleep deprivation-induced mitochondrial damage in the hippocampus neuron. However, acute nicotine treatment ameliorated the REM sleep deprivation-induced mitochondrial damage in the hippocampal neurons. Thank you. If you have any question, you are welcome to ask. This is my reference. Thank you very much.